because it's here not so loud. And if you think you have connection problems, you switch off your video again, because anyway we more or less only see your presentation, and the video is not so important then anymore.
Yeah, I will do it now. Okay. Great. Okay. Okay, shall I start with the talk?
Maybe you go closer to your microphone, please.
You don't hear me very well.
We can hear you, but it's not very loud, and we have set everything to maximum sound now already.
Okay, the microphone is actually on the camera, so I cannot go closer. Is it better now?
Yeah. Yeah, it's better.
Okay, that's great. So I will start with the talk.
And go to full screen.
Okay. So okay. So the topic of my talk will be the application of carbon nanomaterials.
Can you go to presentation mode with PowerPoint, please?
Yes. Yeah, I'm on the presentation already. But can't you see the transparencies?
Yeah, but not in the presentation mode. We see it in the working mode in the moment. We see your whole screen.
But I am in the presentation. Okay, yeah, you're right.
Okay. Yeah, now it's good.
I forgot this. The most important.
Okay.
So, shall I start? Is it okay now? Is everything okay? Can I start?
Yeah.
Okay. So the topic of my talk will be the application of carbon nanomaterials in electrochemistry, especially in electrochemical sensing of molecules with biological interest. The question now is, why do we use such materials for electrochemical sensing? First of all, because of their conductivity. They have huge conductivity, and they improve the currents, the redox currents of systems and of redox systems, and they are quite stable during the measurement of the redox systems. They have the possibility to improve significantly the Faraday current during the measurements, and they decrease the overpotential of compounds, which means that they help from oxidizing these systems relatively easy. So they lead to an increase of the sensitivity, or better, a decrease of the detection limits of the electrodes, which means that they make the electrodes somehow more sensitive to detect these compounds. So we can also improve much more their electrochemical properties upon modification of the carbon nanotubes, or upon oxidation with several oxidating agents. I will shift first to the first step of the talk, which is the production of these materials. How do we produce these materials? So we use as a substrate a silicon oxide wafer, and we grown on these films the carbon nanotubes by using the chemical vapor deposition, the pyrolysis technique, upon decomposition of various carbon materials, and of course of doping materials. If you wanna dope the carbon nanotubes, for example, for nitrogen doped carbon nanotubes, we use as a doping material acetonitrile. For boron, we use boric acid. For phosphor, we use triphenylphosphate phosphor and so on. All experiments, the synthesis has been done in the temperature range from 850 to 900, and we have used as catalyst in all cases ferrocene. Yeah, as I've told you before, we produce 4 different kinds of carbon nanotubes. The classic, the pristine carbon nanotubes, which are the nanotubes, they don't include any doping element, and they are produced upon decomposition of benzene. And we produce as well the nitrogen doped carbon materials, which they can produce upon decomposition of acetonitrile with acetonitrile work at the same time as carbon source material and doping source material. The third type of carbon nanotubes which we produce is the phosphor doped carbon nanotubes. We produce them upon decomposition of cyclohexane and triphenylphosphate phosphor. And the last type of carbon nanotubes which we have produced is the boron doped carbon nanotubes, and they have been produced upon decomposition of ethanol and in the presence of boric acid. As I've told you, in all cases, we have used as catalyst ferrocene. This is a schematic presentation of the equipment which has been used for the production of carbon nanotubes. You can see here, this is the oven which we use. This is the reactor on the furnace, which leads to the furnace, which is introduced to the furnace. From this position, we spray the solution, I mean the carbon source material and then doping source material with the catalyst is sprayed, and with the help of a current gas is shifted to the oven, which the decomposition takes place. Yeah, and after cooling up of the oven, we collect the chips which have the film, which have on the surface the carbon nanotubes. Yeah, this is actually the same point which I showed before. I mean, we have these possibilities. Either we use only a carbon source material and we get pure carbon nanotubes, or we use a doping material, in this case acetonitrile, and we introduce also nitrogen, we get a nitrogen doped material. Yeah, if we use instead of acetonitrile triphenylphosphate phosphine, we get instead of nitrogen phosphate doped material, and if we use, for example, boric acid, we get a boron carbon nanotubes. Yeah. So the second step is to, okay, when we create them, we try to modify them to improve their surface first, to increase the electroactive surface and also their activity as electrochemical activity as electrodes. Yeah. So we start first to oxidize them by using standard oxidizing agents like hydrogen peroxide, nitric acid, also ozone has been used for this. We produce on the surface upon oxidizing several oxygen-containing functional groups like carboxyl or carbonyl and so on. And we have found that upon oxidation, with the oxidation, the electroactive surface area increases. So we have a better area for oxidizing the redox system we are interested for. And so we get more product current, oxidation and reduction current. We had also some problems upon oxidation. We observed that the kinetic of the redox system becomes a little bit slower, yeah, which means that this is a kind of disadvantage from this modification. We have tried something else recently to decorate the carbon nanotubes with some metallic nanoparticles. Some metallic nanoparticles like, for example, gold, silver, iridium, and so on. This was a project in collaboration with the working team of Professor Wildau of the Technical University Ilmenau. They produce especially this kind of metallic nanoparticles of wished size. You can select the size with your own and they can produce it. So we have observed that with the decoration, of these carbon nanotubes with metallic nanoparticles, we increase significantly the active electrochemical area, which means we get better currents, more currents upon oxidation of the redox system, and we decrease the transfer resistance of the whole system, which means that we increase the kinetic upon modification with metallic nanoparticles, and we have observed in general that the electrochemical quality of the carbon nanotubes increases significantly with the modification with the nanoparticles. So this is an important tool to go further to apply the carbon nanotubes for electrochemical sensing. I mean the carbon nanotubes which have been modified with metallic nanoparticles. So an important step in our research is also the characterization of the carbon nanotubes. Yeah, we before we use them for electrochemical sensing, we have to characterize them. We have to see whether we have carbon nanotubes and whether they are modified correctly, yeah, with the metallic nanoparticles. For such method, for such characterization, we use the scanning and the transmission electron microscopy in combination with ADX and XPS methods in order to identify the art and of the metallic nanoparticles, which are to make an elemental analysis somehow of the carbon nanotubes. We combine also this method with Raman spectroscopy method in order to investigate the defects which are produced upon modification. Yeah, I show now some representative images, same images of the pristine carbon nanotubes which they recorded. You can see that the carbon nanotubes are free from amorphous carbon. I would say quite clear, and they are not well structured, something which is expected because of the pristine, they have this known structure, and their length is about 16 to 18 micrometers. The 10 images reveal that they include some rest of the catalyst as iron nanoparticles. You can see them on these black points inside of the tubes. They have an outer and inner diameter of about 80 to 100 and 5 to 10 respectively. And they are quite different, the structure of the pristine nanotubes compared to the nitrogen top carbon nanotubes, which are quite altered, and they have great length. Their length lies at about 20 to 30 micrometer. They have, as was expected, the so-called bamboo structure, which can be seen from the 10 images. These are the 10 images of the nitrogen dot carbon nanotubes. They look like bamboo. They include about, we calculate 7% of nitrogen. And they have as well somehow rest of the catalyst, iron catalyst, but significantly less compared to the pristine carbon nanotube. Yeah, they have an outer and inner diameter in the range from 80 to 110 to 20 respectively. So the structure of pristine of phosphor dot carbon nanotubes, it's more or less between the structure of pristine and nitrogen. Their orientation is relatively good, but bad compared to the nitrogen. And they are quite shorter compared to nitrogen. Their length lies at about 10 to 12 micrometer. They are also anyway clean from amorphous carbon, but they are highly topped with rest of the catalyst. All this black powder which you can see here is rest of the ferrocene, which lies yet now as iron metallic nanoparticles. Yeah. We estimate that the amount of doping material in the carbon nanotubes lies at about 10. Completely different structure poses the the boron carbon nanotubes. Okay, they're quite short. Their length lies at about less than 1 micrometer. They are the shortest carbon nanotubes we have produced. They have a very, very good orientation, and they are filled with bore oxide. These insides, everything is bore oxide. They have also a head with bore oxide. They include about 7% of boron in totally, and they're quite big. I mean, they have about 100 to 200 outer diameter and 80 to 180 inner diameter. So it is very interesting that when you treat them with hydrochloric acid, the bore oxide is eliminated, and you get this kind of structure. You get a kind of macaroni type carbon nanotubes, which are quite empty, and you can either modify it or introduce something inside and use them for example, for electrocatalyzing some redox system. These are some representative 7 and 10 images of carbon nanotubes, actually boron carbon nanotubes, that are modified with gold nanoparticles. The white points you can see are the gold nanoparticles, and you can see that upon the modification, the metallic nanoparticles are homogeneously dispersed in the whole carbon nanotube area will be modified, and no agglomeration takes place. Also, we can clearly see that with the modification with metallic nanoparticles, we don't have any changes in the structure of the carbon nanotubes. So this is some ADX analysis of the metallic nanoparticles which are lying on the carbon nanotube. With this measurement, we prove that the particles which are lying on the surface are gold. Okay, an important step is also the electrochemical characterization of these carbon nanotubes which we have produced. And for the electrochemical characterization, we have selected a standard redox system which is relatively fast, that means reversible, and this is the hexacyanoferrate 2,3, and reacts relatively fast and is quite stable during the measurement. So this is actually the best redox system which can be used for characterizing novel electrons. And for the electrochemical sensing, we have used standard electrochemistry methods like cyclic photometry, DPV, differential photometry, electrochemical neutron spectroscopy, and amperometry. So for all measurements, we have used a 3 electrode system, which means that we have also always the working electrode was the carbon nanotube based material electrode, which we connected on copper wire, and we have used as counter electrode in all cases a plug-in plate, and all measurements were performed around against silver chloride KCl saturated solution. The cell which I have used for measurements can be shown in this picture. It's quite small, which means that it leads to short distance between the electrodes, especially the working electrode and the reference electrode. Important for the electrochemical measurements is the electrical conductor. The electrical conductor has to be correctly, and otherwise you can have some resistance problem which they disturb the electrochemical measurement. For that reason, I have used for conducting the wafer. This is the silicon oxide wafer, which has on the surface carbon nanotube. I have used a plug-in wire, which I conducted with the wafer with the field by using a silver paint. and I let it dry for 1 day, and afterwards I cover the whole area with a varnish cladding. With this step, you avoid that the silver or the cladding work as well as electrode. So we want that only this part should work as electrode, as a working electrode. And afterwards, I have used this as a working electrode for detecting this system in an aqueous solution of calcium chloride with this concentration. I show now as a representative example the CVs which have been recorded in Bohr carbon nanotubes. For other systems, they look like almost the same. So I did standard measurements like, for example, recording this system, series of this system at various scan rates, and I observed actually a reversible and diffusion control process which was expected for this redox system. So I prove actually that my electrode reacts with the response of my electrode is quite fast. I have done the same with different concentrations of the redox system in this range, and I have observed also that with the increase of the concentration, the current increases. So we have a linear variation of the current with the concentration. The system is completely reversible, and we have a huge rate of electron transfer. And compared, for example, of Bohr nanotubes to the pristine without doping, we have found that with doping of the carbon nanotubes, the kinetic of the system increase. Okay, this is the linear variation of the concentration with the current. The same has been confirmed by using the impedance spectroscopy method. So we observe that we don't have a significant variation of the chance transfer resistance with the concentration, and we observe that the chance transfer resistance decreases significantly, almost 2 times, with the doping of carbon nanotubes. And just for information, I will say that the pristine nanotubes is much smaller compared to the traditional electrodes. You can see what is the advantage of using carbon nanotubes as electrodes. Yeah, I summarize all the results in this table. So we have here the 4 types of carbon nanotubes, which I've produced: the pristine, the nitrogen, the phosphor, and the boron. Doped carbon nanotubes. You can see that the faster kinetic and the less chance transfer resistance has been observed for the nitrogen doped carbon nanotubes. Yeah, and then the more worse, I would say, for electrodes is the pristine carbon nanotubes, which they do not have any doping material. But just as I told you, the pristine carbon nanotubes work much better compared to the traditional electrodes. So once more in histogram, the results, you can see that upon doping from unpristine phosphor, bore, and nitrogen, the barrier of redox reaction decreases, and which means that the chance transfer increases, and which means that the kinetic of the system increases from the pristine to the nitrogen. And once more here, a table which shows the electrochemical parameters which is calculated for the redox system. DL is the limit of detection, and sensitivity is the sensitivity of the electrode against the redox system. You can see that the lower detection limit and the greater sensitivity has been observed for the nitrogen doped carbon nanotubes. Once more in histogram, you can see that the limit of detection tends to decrease from unpristine to nitrogen, and consequently the sensitivity increases from the pristine to the nitrogen, which means that the doping with nitrogen leads to better electrocatalytic activity. A short investigation of the effect of the temperature has been also done. And we observe expectable results that the kinetic is improved somehow with the temperature, something was expected. The same with impedance spectroscopy. We have an increase of about 60% of the kinetic of redox system when we increase the temperature from 5 to 30 degrees. And the same, we have a decrease at about 45% of the barrier of the chance transfer resistor if we increase the temperature. Okay, and we also observe that upon increasing the temperature, the exergonic reaction shifts to the direction of the iron-free, of the redox system. A short investigation also of the concentration of supporting electrolyte has been also done, and okay, the results are also expectable that upon increasing the oxidation, the concentration of the electrolyte, we observe a faster kinetic of about 75%. The same result has been observed by using the impedance spectroscopy technique. We have found that the chance transfer of this resistance decreases significantly with the increase of the concentration of supporting electrolyte, which means that for better electrochemical measurement, we should have to use a concentrated KCl solution. Yeah, you can see also here that the chance transfer resistance of the barrier of the reaction of the electrochemical reaction decreases up to 90% if we increase the concentration of the KCl from 10-3 up to 0.2 mol per liter. Also, the kinetic increases significantly, I would say, with the increase of the concentration of the supporting electrolyte. So please note that when you use such systems, you have to increase the concentration of the supporting electrolyte in order to get a reversible behavior. The electrochemical analysis of the standard redox system has been also done with the presence of the metallic nanoparticles. That means that we modify the nitrogen carbon nanotubes and the other carbon nanotubes with metallic nanoparticles, and we investigate the effect of the modification on the electrochemical behavior of the carbon nanotubes. As an example, I show now the measurements which have been done on the gold nanoparticles with various diameters in the range from 5 to 35. We record the cyclic volt nanograms for the three systems, and we have observed that in all cases, okay, we have a reversible behavior, but we have observed that the kinetic of the redox system becomes faster, up to 300%, when we increase the diameter of the gold nanoparticle, which means that the size of the gold nanoparticles, of the metallic nanoparticles, is also important. You can see that the Faraday current increases up to 25% if we increase the diameter of the gold nanoparticles from 5 to 35 nanometers. Once more, the graphical representation of the variation of the current, the anomic current with the diameter of the gold nanoparticle, and the same with the detection limit of the electron. You see that with the modification of carbon nanotubes with gold nanoparticles having a diameter of 35 nanometers, we observe a decrease, or better, an increase of the sensitivity of about 90% of the sensor. The same has been confirmed by using electrochemical impedance spectroscopy. You can see that the barrier of the redox process is decreased significantly by using gold nanoparticles having greater diameter. The temperature effect has been also investigated. We have found also that the temperature helps somehow for the kinetic of the system, something which is expected, and also the temperature decreases the barrier for the redox system. And that is the reason, of course, that the kinetic increases. We went further and we changed gold with other kinds of nanoparticles like silver, platinum, iridium, palladium, and rhodium. As I told you, this material have been produced by the working team of Professor Kula of the Technical University Ilmenau. So we did this work in collaboration with this team, and we tried to investigate the effect now of the art, of the kind, of the type of the metallic nanoparticle of the electrochemical behavior of the carbon nanotube. And we have observed this behavior. The detection of limit tend to decrease from rhodium to gold, which means that within all the metallic nanoparticles which we investigate, it seems that gold reacts better, helps better for improving the electrocatalytic activity. And also the gold helps for the kinetic. From all the metallic nanoparticles which we have investigated, the kinetic of the redox system which has been investigated tends to be better on gold compared to silver better than on platinum and so on. After all of this characterization, we have applied the carbon nanotubes modified with metallic nanoparticles for electrochemical sensing. For this study, we have selected the nitrogen-doped carbon nanotubes, which are the best carbon nanotube material from those which are produced, and we modified with gold nanoparticles of the size which were already investigated and worked better. So, for the electrochemical analysis, we have selected at the beginning 3 important biomolecules, the ascorbic acid, the uric acid, and dopamine, which they have almost similar electrochemical process. The electrochemical process which takes place for all of these molecules, it leads to 2 electron and 2 proton transfer. We have tried at the first step to oxidize all of these compounds in one step in the same solution on nitrogen-doped carbon nanotube without gold. So we add at the beginning ascorbic acid, and we observe a peak at about 0.2, which is caused, which is attributed, of course, to the oxidation of ascorbic acid. And then we add dopamine in the solution, and we observe that dopamine, the peak of dopamine lies very close to that of ascorbic acid at about 0.3. So the 2 peaks are overlaid. And then when you add uric acid, which lies at about 0.5, you can observe a second peak, which is attributed to oxidation of uric acid. So we cannot clearly actually see 3 peaks, 1 peak for ascorbic acid, 1 peak for dopamine, and 1 peak for uric acid. We see actually a peak of uric acid and a peak which is attributed to both of these, which means that this system, nitrogen-doped carbon nanotubes, cannot be used for the simultaneous analysis of all of these compounds. So we have done the following. We modify them with gold nanoparticles. We take this material and we place gold nanoparticles on the surface, and then we observe this behavior. We observe a peak for ascorbic acid, a peak for dopamine, and a peak for uric acid. And it's important to say that the potential now for oxidation of ascorbic acid is shifted to the negative area, which means that it lies at about 0.05, I would say, instead of 0.2, and this leads to a good separation of these both peaks of ascorbic acid and dopamine. So we can say that on this material, the simultaneous analysis in one measurement of these 3 compounds can be successfully done. Yeah? And we have done the same by using the PFV, differential transportometry methods, with a different kind of gold nanoparticles with different diameters, and we have observed that the maximum, the great potential difference between dopamine and uric acid lies at about 250 by using the 14 nanometer nanotubes, and for the greater potential difference of between ascorbic acid and dopamine lies in about 300 with nanoparticles of size of 35 and about 290 with nanoparticles of 14, which I think this is enough for their simultaneous analysis. So we have investigated further the effect of concentration of dopamine in the oxidation of ascorbic acid and uric acid, and we have increased the concentration of dopamine up to 20 times greater than that of ascorbic acid and uric acid, and you can even see that when the concentration of dopamine is 20 times greater than that of ascorbic acid and uric acid, you can even still see the peaks of the both biomolecules, which means that the ascorbic acid and the uric acid can be detected, can be analyzed in the presence of an excess of dopamine. The same has been done, the effect of concentration of uric acid on dopamine. You can also here see that the dopamine peak can be detected even if the concentration of uric acid is about 20 times greater than that of dopamine. So I summarize all the results in this table. You can see from these results that by using gold nanoparticles and modifying the carbon nanotubes, you can reach a negative overpotential of ascorbic acid, which leads to a greater peak separation between ascorbic acid and dopamine, and of course to separated oxidation peaks between the three biomolecules, which is important because this helps for measuring all three compounds in a single measure. So you can see once more the variation of detection limit of dopamine in the mixture with ascorbic acid and uric acid by using various nanoparticles. The first is the unmodified nitrogen carbon nanotubes modified with rhodium, palladium, iridium, platinum, and gold. The same with the variation of uric acid and the detection of the uric acid, and the same the variation of detection limit of ascorbic acid. So in all cases, we observe that gold nanoparticles is the best material on modifying the carbon nanotube to nitrogen carbon nanotubes for detecting the biomolecules. Important is also a comparison of the extracted data which we got in our laboratory for our novel sensors with those published in the literature for other novel sensors containing as well carbon nanomaterials and metallic nanoparticles. First, a comparison of the extracted data with dopamine. So our detection limit of dopamine in nitrogen carbon nanotubes modified with gold nanoparticles lies at about 4 times lower compared to other electrodes like classic carbon electrode modified with a polymer, or 13 times better compared to poor gold electrode. Is much better compared to single wall carbon nanotubes or to plug-in or other standard electrodes. The same with uric acid. The extracted detection limit seems to be much better compared to other standard electrodes or novel electrodes published in the literature. And the same also for ascorbic acid. The obtained detection limit seems to be much better compared to published unpublished novel electrodes. Application for further application on other biomolecules is also possible. I have tried, for example, to oxidize glucose on the nitrogen carbon nanotubes modified with gold nanoparticles without the enzyme which is required for the oxidation on glucose, and I have observed a peak at about 0.2 against silver chloride of glucose for the oxidation of glucose. And I add as well uric acid in the solution, and I try to detect uric acid in the presence of glucose, and I have observed a second peak, which means that uric acid can be measured in the presence of glucose, and glucose can be measured in the presence of uric acid. But as I tell you, it's important that glucose can be oxidized in the absence of the present, in the absence of the enzyme, which is required for oxidation. Further application are acetaminophen, which is the paracetamol in the presence of N-acetylcysteine, also have been measured both, and we observe separated peaks, which are well separated, who are not overlaid, and which means that you can measure them both in a single measurement. It's also important to mention the effect of on the current of the decoration with growth nanoparticles. You can see that with the modification with growth nanoparticles, an increase of about 4 times of the Faraday current you can observe. This is a cyclic voltammogram which has been reported on this system for glucose. So this shows the importance of modifying the carbon nanotubes with growth nanoparticles. I have tried actually to measure a force system, redox system by using nitrogen top carbon nanotubes in the presence of gold nanoparticles, and the result is the following. The results are the following. First, uric acid, various concentrations in the presence of glucose, and in the second step, ascorbic acid. So a third peak has been observed, and then I added dopamine, and I observed a fourth peak. So it's possible somehow to investigate by using this kind of materials for redox system in a single measurement. But these experiments are still in process, so I cannot present some more results. I think important should be also the diameter of the particle in order to separate all of these peaks. But as a first step, I am quite satisfied for this measurement. Okay, let me now make a summary of my talk. I'll try to be as short as I can and as clear as I can. So I have produced these 4 kind of materials: the pristine, the nitrogen top, the phosphor top, and then boron top. And I have investigated them electrochemically, and I have found that these kind of materials, actually the nitrogen top material, work better as electrochemical sensor. The nitrogen top carbon nanotube has been modified with several metallic nanoparticles, and it has been found that from these metallic nanoparticles, the gold nanoparticles work better in electrochemical sensing, and this material has been used for electrochemical biosensing of these 3 biomolecules: ascorbic acid, dopamine, and uric acid. And it has been found that all of these 3 compounds can be measured, can be detected in a single measurement on carbon nanotubes, nitrogen top modified with gold nanoparticles. A further analysis can be done by using a 4 quaternary system, including as well recorded on the same system, which is quite promising. And finally, a comparison of our extracted detection limit obtained on nitrogen top carbon nanotubes modified with gold nanoparticles compared to the literature seems to be much better in our cases. Yeah, in closing my talk, I would like to thank the college of me here in Ilmenau, also in Dublin, in Poland, in UK, in also my PhD students, Mr. Otan and Mr. Taha, for their help. And yeah, I would also like to help you thank you for your attention. And I don't know now if we have some time for questions. I would be glad to get some questions for you.
Yeah, thank you, Dr. Tsierkezos. Please, questions from audience. Come close. Just give us one moment.
Okay, I don't know if we have still time. How much time do we still have for?
I think we have approximately 10 minutes.
10 minutes, okay.
Hello. Thank you very much for your interesting presentation. I would like to ask about the problem of electrode fooling. So, how you clean your electrode from the one measurement to the following one? Or you consider it as disposable?
Okay, actually, I forgot to tell in my talk present in my talk that this is a kind of one-use electrodes. They're quite cheap, so we use them just for one single measurement, and then we waste them. I mean, we produce several electrodes for the measurements, and for each measurement we use single electrode, and then we waste it. We don't have to clean it. Each electrode is used can be used for a single measurement.
What about, thank you, what about reproducibility of your measurement?
It's quite good. It's quite good, I would say, because the experiments which I have presented have been repeated a couple of times, and the results are quite same. I mean, it's quite reproducible.
Mm-hmm. And other question about wettability properties of this nanotube forest. Can you tell if a drop of water we put on this on the surface of nanotube, if it's full of pore of this forest or not? Did you make some such measurement?
I didn't understand the question. I mean, we measure actually
wettability properties about because you work with the water with the aqueous solution. So I ask you if water penetrate into between nanotubes, because you use like a forest of nanotubes. So if water penetrate to the substrate through this nanotube forest, literally speaking, forest.
And what kind of effect do you have in the
wettability.
Okay. So,
wettability, you work with the aqueous solution. So if water cannot penetrate through all this forest to the substrate, so it's it's a question. So
I have to take some 10 images after the measurement probably, because after introducing, you mean the fields in the solution.
Wettability properties of this structure of a forest with nanotubes. It's if you use aqueous solution, if the water penetrate into this between these nanocarbon nanotubes, is it full pore of this structure or not?
If you mean if water is introduced in the structure of the carbon nanotube, inside of the tubes, you mean?
Not inside of the tube, between the tube, because you use a tube like a forest, vertically arrayed.
This is something which you cannot avoid. I mean, it's introduced in aqueous solution. How can you avoid this?
If the water contact with the surface of the nanotube, or it's only contact partly or full, you can reach full contact of the water, because electrochemical surface active area will depend on the wettability properties of your nanotube.
the carbon nanotubes are homogeneously dispersed on the surface of the silicon wafer. There's probably some holes somehow in the surface, but they are actually orientated on the wafers. There's like a carpet, and but I think in some cases, in some position, probably there are some holes, which water can be, but.
You told that the dimensions of the nanotube is about 15 micrometers. So it's they oriented vertically. So if you put, my question is, if you put water, it does not mean that water penetrate into this 15 micrometer, or just it's, for example, to 5 micrometers, or it's will be equal to the height of the nanotube, or not?
I mean, the length is about 15, you mean? The length of the nanotube. The length, you mean?
Yes, the length, but the length determines the thickness of the carbon nanotube layer, if I understood correctly, because it's vertically alignment.
Yes.
Yes. So.
Okay, we can.
Okay. So maybe because I'm afraid that we limit in time.
It depends on. Is there any other questions?
Okay,
Yeah, please.
Thank you very much. Thank you.
I wanted to ask about toxicity of nanotubes for just organism. How toxic is it or not? How it influences biological systems in a wrong way, I mean.
I didn't hear the question. Can you please repeat one more the question?
Yeah, and
Okay.